Hello again, everyone. My name is Austin Craver, and I'm back again to give you a little bit more instruction for your Biology 105 online lab. The topic I would like to address in this video is a little bit of guidance on how to perform the lab reports. Now, as I stated in the syllabus video, there is a rubric for the lab reports provided on Canvas, and I'd like to show you where that is. So this should be your Canvas home screen. Over on the left, there'll be a link called Assignments. If we click on this, it'll give us a link. It will give us a page that has all of the assignments for the semester laid out. Now if we click on one of the lab reports, at the bottom there will be a long list of all of the points that we're going to be grading you on when your instructors sit down to grade your lab reports. And here I've made it a little bit larger to make it a little easier for you guys to follow along, because I'm going to go through this rubric and go through the steps of a lab report, the different sections of a lab report, to give you a little bit of guidance on how to write lab reports. So first off, I want to make sure that it's clear. In the process of writing a lab report, you're making an argument about how something works in the real world. And the process of generating a report is going through a series of codified steps to both convey this information that you discovered while performing an experiment, as well as to create an argument about how you feel or how you think the world works based on the information you've had access to by performing your experiment. Now, lab reports will be broken down into several sections. An introduction that will include your observations, hypothesis, predictions, and question. A method section that will lay out what you guys did in the lab, where you will explain to the instructor exactly what you did. A results section where you will present the results of the experiment that you performed. And a conclusion section where you will take the results, tie it back into your hypothesis to give meaning, so that you can assess whether you feel like your hypothesis was supported by the data you found or not. So, let's start with the introduction. The main goal of the introduction in a lab report is to give the reader the necessary background information. You will be responsible to show your understanding and to provide understanding to the reader of the relevant biological facts for each lab. This is covered by providing sufficient background information, your observations, and previously performed research points on the rubric. While writing the section, I heavily encourage you to rely heavily on your learning area, and to be sure to cite the sources that you utilize, such as the learning area. On the topic of sources, I also want to stress citing .com websites is completely inappropriate. If you have any doubt about what sources are valid, please contact your instructor and ask what their preferences are prior to using any source. So, after you provide the reader sufficient background so that they can understand the important biological components that are going on for that particular lab, you will then move into asking a question. Fundamentally, every experiment that's performed in science is to attempt to answer a question. These are what we would call your research questions. And they're going to be the elements of the world that we're trying to figure out with that particular lab. Each lab is going to have a different question, and the learning area is going to provide you a lot of guidance on how to write good questions. The things that I encourage my students to keep in mind for this is when you're writing a question, be sure to be very specific. Also, make sure you're objective. Remove any subjective terminology. In science, we're not worried, and we don't concern ourselves with the subjective world. The reason being is because subjective information is different from individual to individual. So in science, we will focus on objective statements and facts, as those are true for everyone. The next thing you'll generate while writing the lab report is to synthesize a hypothesis. Now, hypotheses are very specific, testable answers to your question. I want to warn my students that as you write your question, your hypothesis, and your prediction, you're going to feel like there's a lot of similarities between the three. The reason being is because there are. Each of these three components are very, very similar information, just presented and worded in different ways to perform a different function in the report. The question will establish what the research question is, the piece of information that the report is targeting. The hypothesis will present a possible answer to that question in regards to how the world works. Your prediction will take your hypothesis and apply it to the specific experiment that you'll be running. So basically, if the world works this way, when I do this experiment, I should see this outcome. Each of these three components are going to have a lot of overlap, so keep that in mind while you're sitting down to write them. Once you generate your prediction, this will act as a bridge between your hypothesis and the rest of your introduction over to your method section, which is where you're going to describe how the experiment was actually performed. Alright, so for the method section, the main goal that you're going to have is to lay out all the technical aspects of how you perform the experiment, and in great enough detail that anybody else could pick up what you wrote, sit down, and completely accurately recreate the experiment that you ran. This is the main topic, or the main goal of the method section. There are also additional materials we want to make sure you guys incorporate into your method sections that you write. First of all, you'll need to discuss appropriate controls for each experiment. Experiments inherently act as comparisons, for the most part, between two units. The controls will give you that baseline to compare against. Now, it's going to be easiest to discuss this if I introduce you to two pieces of terminology, and that's an independent and a dependent variable, because these are the basis for how experiments are designed. Experiments are designed by creating generally a minimum of two units that are identical, except for the independent variable. For the first lab that you guys will perform, you're going to be investigating the validity of horoscopes. In that case, we broke all of the students up into two groups, and we gave one set accurate horoscopes and one set inaccurate horoscopes. The accuracy of the horoscope is the thing that we changed in between these two groups, and that's really the root of the question that we're asking for this first lab. So the independent variable, the thing that's different in between the two groups that we set up, is, like I said, the accuracy of the horoscopes given to the students. Besides that, the two groups are treated identical. That way, when we compare the results, or the dependent variable, from each of those two groups, we can create a comparison and assess that one is different from the other in some way. This will be crucial as you guys generate your arguments for or against your hypotheses in your conclusion section. We will also ask you to address if the experiment was replicated. Generally speaking, scientists are not concerned about how something works once in one place, but instead we want to learn about how things work in the world or the universe as a whole. So, to try to access this information, we always replicate our experiments. For most of the experiments you'll perform over the course of the semester, each member of the group will be replicating the experiment in its entirety on their own. You want to make sure you address this in the text of your method section. You also want to make sure you address how you measured your variables, which is basically how you assign the independent variables if you had the opportunity for that particular lab to assign those independent variables, as well as how you measured the dependent variables, or the result from your experiment. This concludes the methods section. The next section that you guys will be working on is your results. In the results section, you guys are only going to be presenting the facts, the data that came out from the experiment. I want to stress, when you're writing the results section, you do not need to give any meaning. In fact, you should not give any meaning to what the results mean. This should be addressed in the conclusion section. The results section will be broken down into several subcomponents, and this can be found in the rubric. First of all, making sure that each lab, or each lab group, produces and represents the raw data is going to be critical. Raw data is going to vary from lab to lab, so be sure to ask your instructor what's expected for each experiment. Most commonly, what it will be are before and after photos of the experiments that you guys perform at home. Make sure that you include this, as this is also going to be a component of your individual lab report grade. This is how you would document that you performed the experiment. You will also have to generate tables and figures. These tables and figures will need to be summary data for the experiments, and your instructor should give you advice on how to analyze and summarize the data, the information that you receive from each experiment, as it will be a little different for each lab. In general, what you guys will do is average all of the independent variable replicates that you perform. Now what's that mean? It means if member A, member B, member C, they all replicate the full experiment, say they have two independent variables, like the horoscope example that I was providing you, they would take their values for independent variable A and average all of them together to generate one value for the independent variable. One dependent variable value per independent variable class. Averages will by far be the most common way that you will summarize the data. Summarized data will need to be represented inside of a table as well as shown on graphs. Bar graphs are the most common form of representing this information. Your instructor should give you some information about how to generate these graphs. Now, a component of generating tables and graphs for your report will also be labeling them properly. You will need a title and axes labels for every graph, and you'll need to label the columns and rows of tables, as well as give tables titles. In addition to the labeling of axes and titles for your figures, you'll also need to include a caption that goes along with each figure. And what I mean by figures are everything in your lab report that is not text, that was not written. So this would include your tables, this would include your graphs, this would also include your raw data that you provided. Each of these will need a very small blurb of text to describe what you're looking at. Be sure to separate this blurb from text from the rest of the writing in your report. Make sure that it stands out and it's clear that it is associated with a figure. In addition to the tables and figures, or tables and graphs that you'll generate for the results section, we also expect you to generate a very brief textual summary of the data that you saw. This text summary should include the main trends that you see in the data. The simplest way to start thinking about this is Group A was greater than Group B. Group B was greater than Group C. And providing those sorts of comparisons, those main overview patterns. You do not and should not repeat the exact numerical values that you received and you're presenting in tables or figures in the text. This concludes what's expected of you for the results section. The last component I want to mention is the conclusion section, as I alluded to at the beginning. This is going to be where you create an argument for the validity of your hypothesis. You will take the results, the facts, the data that you guys pulled out in your results section from your experiment. You will analyze their meaning, and you will present how they influence your hypothesis. The basic flow of this should be something along the lines of, in my experiment, this group was different than that group. This means this for my hypothesis, and here's why. While writing this section, you will be judged on the accuracy and clarity of the logic and the argument that you'll make for your hypothesis. Be sure to be incredibly thorough. Do not expect the reader to infer any of your logical thought process. You will need to guide them and provide the information yourselves. This is the main component of your conclusion, but there are also additional topics that we'll have to address using the conclusion section. The primary one, well, the next one being, is a critical analysis of the experiment you ran. So I want each of you to think about what may have been good and what may have been bad about the experiment you performed. And if anything in the methods section could bring the validity of the experiment into question, I want you to address it in the conclusions. Basically, think about if somebody else was going to try to argue against you, you're trying to guess what their argument would be and present it to the reader before somebody else argues it against you. I would also ask that you then go forward and address these errors, solve them. So you would provide potential revisions for the experiment if it were to be performed in the future, and how those errors or biases could be removed. Errors and biases will vary from lab to lab. If you have questions or need assistance with this, as always, ask your instructor. Now, the last component of the conclusion that you need to make certain you include is comparing your results to previously published work. This will require a citation. You can use the learning area and the pre-lab information as previously published work, unless your instructor tells you otherwise. You'll need to reference how your conclusions for your hypothesis fit and match or don't match with what information's been found by other scientists. And, like I said, the learning area is a representation of information that's been found through other science. This concludes the main grading components of your reports, but there's a few other general guidelines I like to suggest that will help you when writing reports for this course. First of all, in my classes, I require that the majority of the contribution from students comes 24 hours before the due date. This rule may vary from instructor to instructor, but in general, it's a very good pattern to follow. The reason being is because it'll give other group members time to look over what you've written. Since everyone's going to be assigned the grade for the group for the lab report, each of you should take a look at what other people write, so that everybody has a voice on every part of the report. Even if you do assign sections to individual group members, please take a step back and go back over it. Make sure you get a decent amount of material done in early enough that other group members can review. Also, looking over each other's work will help make sure that the elements that occur through multiple components of the report are kept consistent. Hypotheses especially occur repeatedly throughout the report. You want to make sure that the person writing the conclusion and the person generating the hypothesis in the first place are on the same page. This concludes my brief rundown of the expectations of reports in this class. If you have any additional questions or questions based on specific labs, please reach out to your instructor either via email or in a Google Hangout.